Welcome to another video tutorial on staining techniques. Let's get started. Okay, so staining techniques. So now, as the name tells over here, staining means coloring. Okay, so yes, so in the previous video we studied most of the microscopes, how do they work and all those things. Now here we will study about the staining techniques. So we stain, that means we color most of the cells and most of the microorganisms and then observe under the compound microscope. Okay, so why? Because cells or microorganisms, no, they appear transparent or colorless when you view under the basic microscope. Because when we are observing under microscope, under the basic microscope, usually we slice them into a very thin sections. Okay, so it will be very thin sections, and apart from that most of it will be composed of water. Okay, as a result what happens is they do not absorb or reflect light in a way that is easily visible to our eyes. Okay, and hence we cannot visualize them when we don't color them or when we don't stain them, and that's the reason we need to stain them. Okay, and we need to observe them under microscope. So for staining purposes we have many different dyes available. Okay, and we also have very many different techniques of staining. Now for example, if you want to observe the entire organism, so you have one method of staining, and if you want only a particular part or particular region to be observed, you have a different staining technique, you use a different stain. So like this, so there are many different techniques and many different stains which we use for visualizing under microscope. Okay, so that is what we are going to study here in this PPT. Staining techniques. Okay, staining methods are used to elevate the visibility and highlight specific morphological structures of the microorganisms. It is also used to preserve them. Yes, so we stain them or we color the specimen mainly to elevate, that is to increase the visibility. Okay, and to highlight the specific morphological structure. Sometimes you cannot see some morphological structures very clearly. Okay, only when you stain them you can see them clearly. And and also for preserving them, we you can preserve them, you can slide, you can color them, you can mount them, you can also preserve them for a long period of time. So for doing all such things we do this staining techniques. Okay, and due to lack of contrast, the structural details of organisms cannot be seen under a light microscope. Hence we use dyes to stain cells. Dyes binds with cellular constituents, producing color contrast and increasing visibility. Yes, and the second thing is there is no contrast. That means you cannot know, you cannot find a very good difference between the slide and the specimen, because as I told, so there will be a very thin section, they will be very much transparent, colorless, and hence you cannot find the contrast at all. Okay, and that's the reason we use dyes. Okay, and these dyes will go and bind to the cellular components, and hence they increase the contrast where you can observe them very clearly. Okay, and then among the various types of dyes, it is the positively charged cationic ones that play a crucial role. Dyes such as methylene blue, crystal violet, saffronin, etc., with their positive charge specifically bind with negatively charged cellular constituents such as nucleic acids and acid polysaccharides, and even the cell surface of bacteria, enhancing our ability to observe and study these structures. Yes, so in stains also there are many different types of stains. Okay, so there are positive stains available, there are negative stains available. Among them, no, positively charged stains, they play a very crucial role and we use them mostly. Okay, so they're also called as cationic ones, that is positively charged ones. So example for positively charged stains or dyes is methylene blue, crystal violet, saffronin, all these are positively charged, and it works on the basis of the opposite charge, that is these are positively charged dyes and they go attract to the negatively charged components of the cell. Now for example, nucleic acids, acid polysaccharides, cell surface of bacteria, all these are negatively charged. So this positively charged bacteria, sorry, dyes will go easily attract to the negatively charged components of these cell or bacteria. Okay, so hence they give you a good contrast. And based on the types and number of dyes used, no, we can differentiate or we can categorize them into different types of staining methods. Okay, so these are the types of staining techniques. So first is simple staining and second is differential staining. So the main difference between both of this is in simple staining we use only one stain. Okay, so based on the purpose we use only one stain here. Okay, and here we use two or more number of stains. Okay, more than one stain we use it in differential staining. Okay, and again in simple staining we have two types, that is direct and indirect. Direct is positive staining it is also called, where we use positive stains, and the second type is indirect stain where we use negative stains. Okay, and coming to differential staining, no, differential staining we can use it for two purposes. The first one is separating into groups. First one is gram stain. Gram stain means now when you take that is bacteria especially, so there are two groups of bacteria, that is gram positive bacteria and gram negative bacteria. So now this gram stain is used or it is done to differentiate gram positive from gram negative, and it gives different color. Gram positive gives different color, gram negative gives different color in the same slide, so that you can know. Okay, so which is gram positive, which is gram negative easily. You're separating into groups here. Okay, like that only. You have acid fast stain. Yeah, there are organisms which is acid fast, which is non-acid fast. Again, acid fast gives you a different color here, non-acid fast gives you a different color here, which you can observe both of it in a single specimen and you can differentiate them easily. Okay, so like this, to differentiate into groups we use the differential staining, and the second purpose is visualization of specialized structures. Now for example, flagella stain is done especially to study the flagella. You stain the flagella there and you study the flagella in detail. Then you have capsule stain, where capsule is stained and capsule is studied in detail, like that. Spore stain, like this special structures are stained over here and they are visualized. Okay, so this is used for two purposes and these are the types of staining techniques. Fine, and then yeah, first we'll start with the simple staining. Okay, yes. Simple staining is a basic microbiological technique that uses a single dye to color microorganisms, making them more visible under a microscope. So this method highlights the overall structure and morphology of microbial cells without providing detailed information about specific cellular components or differences between types of cells. Okay, so as we discussed in the previous slide, no, in this simple staining technique, we use only one single stain. Okay, only one single stain, and mainly this is one of the simplest technique, and mainly we do this to visualize the morphology and the structure of the microbial cell. Okay, you don't need to study anything in specific over here. Just you want to know how is the arrangement of the cell, what is the size of the cell, what is the shape of the cell. Okay, and how does it look? You basically want to know how does it look. So that time we use this simple staining technique. Okay, see, the main purpose of it is to determine the shape, whether it is spherical shape, whether it is rod shaped, whether it is spiral shaped. Okay, to know this we will use the simple staining technique. Apart from that, to know the size, that is relative dimension of the cell, we use the staining technique. And then to know the arrangement of the cell, whether it is present in chain, whether it is present in groups, clusters or pairs. To know that we will use this simple staining method. Okay, and as I told, this is one of the simplest method and it is very quick and you can observe very quickly, and you can come to a conclusion what is the morphology of the cell. Okay, and then okay, now coming to the procedure of simple staining, it is very simple. That is, smear the small microorganism sample on the glass slide and air dry. Heat fix the slide to adhere the cells to the surface and kill the microorganisms. Yes, so now the first step what you do is you'll take a clean glass slide. Okay, and then so using a loop you're gonna take the small amount of the microorganisms sample and you have to make a very thin smear on the, see, this is called as smear, this is a glass slide. By using a loop you have to make a very thin smear on the slide. Okay, so only when it is thin you can observe the structures very clearly, and that's the reason you need to make a very thin smear. After making a thin smear, you're gonna heat fix them. So how is that you heat fix? Is that you're gonna take this slide and just um, you're gonna place it on the burner. So, only for a second, only for seconds. It's not completely placing it on the burner. You're gonna just bring it on top of the burner. So only for 1 second. Move it away. Again bring it. Again move it away like that. So not continuously, but bringing close and away, bringing close and away like that. You're gonna heat fix it for few seconds. Why do we do that? So when we do this, so the cells are gonna adhere onto the slide. Adhere in the sense it's gonna get fixed onto the slide. Okay, and that's the reason we are gonna heat them. Okay, and then simultaneously it's gonna kill the microorganisms also. And apart from that, next step is apply a single stain such as methylene blue, eosin, crystal violet or saffron to the smear for a specific period. So after heat fixing, now you're gonna flood the stain onto the smear. You're gonna put just gently you're gonna put the stain onto the smear. Okay, and allow it to remain for few minutes. Okay, for around some 2 to 3 minutes you're gonna leave, and then gently you're gonna water wash to remove the excess stain which is present. Okay, and then you will observe them under microscope. See, you will wash them, you will dry them, leave for 2 minutes, it's going to get dried, then you're going to observe under microscope. So this is a simple method, simple staining method which we are going to follow. Okay, so take a slide, smear the sample, heat fix them, apply the stain. Okay, wash the excess stain, air dry, and then you're going to observe under microscope. Okay, so and here we use many different stains like methylene blue, so highlights bacterial cells and is commonly used in bacteriology. As the name tells here, it will give you a blue color. And saffronin produces reddish pink color and it is often used as a counter stain but works well for simple staining also. And then next is crystal violet, a purple vibrant stain that provides excellent contrast. Like this, there are many stains, and many stains gives you different colors. Okay, and they have a different property as well. Okay, then next is positive staining. Positive staining involves using basic dyes that have positive charge which binds to the negatively charged components of the cell, effectively coloring the cells themselves. The specimen is typically heat fixed to a slide before staining, which helps the dye adhere to the cells. Okay, so that is, see, as you can see in the picture over here, so this gives you a very good picture. So wherein this is the dye which we are using for staining. So the dye is actually positive in nature. Okay, so they are basic dyes, they are positive in nature, and in the cells now most of the components are negative. Okay, so when we stain this positively charged dye onto the cell, when we put this onto the cell, what happens? The positively charged dye attracts the negatively charged components of the cell and they get colored. See, as you can see here, they bind. See, all the positively charged dye is getting bound to the negatively charged components of the cell, and as a result, so the cell gets colored. So this is a basic simple principle which the positive stain uses. Okay, and as you can see here in the picture, so how nicely the cells are colored here. The microorganisms here are colored. Okay, and some of the common examples for positive stains are crystal violet, saffronin, methylene blue. These stains are often used in techniques like gram staining. We also have differential staining which we are going to study further. So not only for simple staining, we also use this methylene blue, crystal violet, saffronin for other differential staining purposes also. Okay. And why do we use positive staining? Mainly to visualize the cellular structures, for identifying bacteria, and for studying tissue morphology we use positive staining. Okay, so and the opposite of it will be the negative staining. As you can see here, here are the microorganisms, so the cellular part is colored, stained, whereas here the background is colored and you cannot find any color in the organisms over here. Okay, that is a difference. Okay, next coming to negative staining. Negative staining uses acidic dyes that have a negative charge which do not bind to the cells but instead stain the background, creating a contrast that outlines the cell. This technique does not require heat fixing making it suitable for delicate specimens that might be damaged by heat. The sample is mixed with a dye and spread on the slide, allowing the background to be stained while the cells remain clear. Yes. So the second type of staining is negative staining. As I told here the organism or the specimen part will not be colored. Instead, the background will be colored. Okay, why this happens is here we will use negative stain, that is the acidic dyes we use here, and these are negatively charged. And as I told, most of the cellular components are negatively charged, and as a result, what happens when the negative stain tries to attract the negative charge over here, they repel. See, now this is the slide, and here is the cellular components. So when the dye, negatively charged dye tries to bind to the negatively charged cell, they repel, and as a result the cell will not get stained. Instead, the background will get stained. Okay, so hence we call it as negative staining. And here mainly we do negative staining for cells which is heat sensitive. Now, as I told in the previous staining technique, that is positive staining, we do heat fixing. So when we do heat fixing, what happens, the microorganisms will get adhered into this slide, and it will get killed also. Okay, but there are many other microorganisms where when you heat fix, no, they're very sensitive. uh, they might be damaged to such an extent that you cannot even visualize their morphology. Okay, and that's the reason we stain in such a way so that the cell is not getting affected. Okay, and the common stain which is used over here is nigrosin, Indian ink. Okay, so here mainly we use them for visualizing structures like capsules around bacteria, morphology of virus, and all those things. Okay, and yes, negative staining is often used in electron microscopy to visualize fine details of cellular structures and is effective for observing delicate organisms. I said, delicate, very delicate. If you heat fix only when their structures get destroyed, no, so that time we do this negative staining. Okay. And then advantages, a simple stain offers several benefits. Firstly, it is quick and easy. It's very quick because you apply only one stain. Within few minutes, it's gonna be prepared and you can observe it very fast. Okay, and minimal steps are present, minimal reagents are required here. That's one advantage. And secondly, it is cost effective. Okay, only one dye you are using here, which is not so much expensive, and it is effective. And then it is ideal for beginners because when you're doing staining techniques, it's necessary that you have to learn how to make a smear, how to make a thin smear, how do you stain. Okay, so in the beginning only, when you are provided with 2 to 3 stains, maybe it will be difficult, so it's better that you learn it with first one stain. So it is ideal for beginners, and it provides, yes, basic visualization to just know what is the cell morphology and arrangement. You can use this method. So it provides a very clear enhanced view of microbial cells, serving as a foundational tool for further microbiological analysis. This is a basic step. So immediately when you bring an organism, you want to know something like morphology and all those things, you can first do simple staining, and after that you can go for specialized staining techniques to know the further details about the organs. That's it.